And now the mystery talk title has been revealed. So some of you are wondering, oh no, how do I get out of this room now that I've seen this talk title very carefully. So sorry for the last minute talk title. It's interesting also somewhat of a last minute talk because I only found out Friday that I'm actually giving 2 talks. So I'm also giving a talk tomorrow, and I just did the foolish thing of looking at the schedule to see when my talk would be. I saw a talk on Monday, and I said, great, talk's on Monday. And I started thinking about what that talk would be. And then as I was thinking about the conference and coming on Friday, and I was like, I wonder what else is going to be at this conference, you know? And then I looked on Tuesday, and my name was there again, you know? So surprise to me. We'll see how it goes. However, I'll just say, without embarrassing the person, that at lunch today, I just informed another speaker that this speaker has a second talk at the conference. So I was not the only one that made this mistake. So this is going to be very different, actually. I noticed that I'm squeezed between 2 hardcore statistics talks, like power and FDR correction and all this sort of really complicated stuff. So I think I'm kind of like the sort of light entertainment in between them with no statistics. Now, when you see evolution, maybe the first thing that comes to mind is species evolution. And of course, that's a really important and well-researched topics, and there's some experts here at CGSI in this topic. But as you can see in the second part of this title, I'm not going to talk about species evolution. I'm going to talk about evolution that occurs somatically. And mutations happen pretty much every time a cell divides in the body. And most of the time, these mutations are sort of inconsequential for the organism. But occasionally, mutations happen in a part of the genome that is a gene that regulates growth and causes that cell to grow quickly or evade the immune system. And then that leads to eventually a tumor, right? So this is a sort of classic example of somatic evolution. But as I said, mutations happen all the time. So you could imagine that, just if you were to measure individual cells in an organism, like in a human, there would be somatic mutations in almost every single one of our cells that distinguish the cell from the original zygote. So you could also think about trying to track just normal developmental processes using somatic mutations. And through a variety of great technologies, which I'll sort of mention a bit, but it's not the genomic technologies section, so I won't spend too much time on that, say that we can now measure single cells from many different cells in a tissue, and then try to reconstruct these past processes, these past evolutionary processes, or this past sort of lineage process. All right, so just to get everyone on the same setting here, with single cell sequencing, and there's different technologies. I'm going to show you one in a few slides. You can imagine that the type of data you get is for each of hundreds or thousands of cells, you measure some locations in the genome where there have been mutations. And the phylogeneticists call these characters for some strange reason. But each mutation is a locus, a character in the genome, and you record here its state in that cell. And here I've shown a very simple character. It's had only 2 states, 0 and 1. So if we then think about how we might recover a tree from this data, we think that each row here corresponds to a leaf of the tree, which is something we measure at the present time. And then we try to infer back in time the ancestral process and the ancestral mutations according to some evolutionary model. Okay? And that's what I want to actually talk a bit about, is what these evolutionary models are and what they might be that are appropriate for these different types of somatic mutation processes. So if we start thinking about single cell phylogeny, there are a few key questions for how we might build these phylogenetic trees. So one question would be, what mutations do we want to use as our phylogenetic characters? Now, in cancer, it turns out that we have a wide variety of choices because mutations occur across all genomic scales, not just single nucleotide changes in DNA, but larger duplications or deletions of regions of the genome, or even duplications or deletions of whole chromosomes, or even wild events where the whole genome duplicates and you get 4 copies of every chromosome instead of 2. So all of these are possible phylogenetic characters, and we have to make a choice as to which ones we might use, or we could attempt to use them all. All right, then the next question, how reliably can we measure these characters? And unfortunately, for pretty much every single cell sequencing technology, there's pretty high rates of missing data and errors. So we need to think about how we might account for that. And then the third question, which is perhaps a question that is not always asked so much, is what model do we want to use to describe the evolution of these characters? And these three questions are all related to each other, actually, because if I say that I want to measure my evolutionary process using duplication and deletion events, then I need a model that describes how duplication and deletion events change the genome, right? So how could we describe these different models? So we're going to actually, for the first parts, to think about sort of single nucleotide mutations, but very simple characters. Okay, and every evolutionary model can be described as something that I'll call the state transition graph. Not really standard terminology, but it's close to the terminology that is used in the literature. So here's a very, very simple example. So here's a tree again, a phylogenetic tree on 3 leaves. What I'm showing you are 2 characters, all right? One character, the first character here, has 2 states, which as a computer scientist, I'll label 0 and 1, right? The other one has 4 states, which as a biologist will label A, C, T, and G. Right? Okay, so now you can see the mutations that I've labeled on the edges of the tree to indicate when there are changes in state of each character, right? So this yellow mutation is when the C changed to an A, that second character, okay? So what model should we use to describe both the allowed transitions and the probabilities or score or weights of these transitions? So we might think of having a very unrestricted model, and this is what's called the finite states model, where we can describe this via a graph, where every node of the graph is one of the states of our character. So here I'm showing you this possible state transition graph for the second character. There are 4 states. Then I'll have directed edges indicating which transitions are allowed to occur over the evolutionary process. Okay, so here you see that I think I missed a few actually. This was supposed to have every possible transition, but I did miss a few directed edges, as you can see. And I've shown you here with labels, again, this C to A transition is this directed edge right here. And if I put weights on these edges, I can turn that into a parsimony type score, which you might have heard about. If I put probabilities on these edges, I can turn that into a likelihood score. Okay. So this is the most classic model. This is the, in a way, model that allows all transitions with different rates or probabilities. There's versions of this that you might have heard of with different, if the rates are very specific, this is called the Jukes-Cantor model for DNA evolution. But we could think of actually doing more restrictive models. And here's actually the sort of opposite extreme, where I have an evolutionary model that has, for a character, only 2 states, 0 and 1. 0 goes to 1, but not the reverse, so that's not allowed. And then there's this other constraint, which I illustrate here with this little, Pac-Man type character, which is that transition is only allowed to happen once over the whole evolutionary history. And this is a very famous model, actually, sometimes called the infinite sites assumption, the no homoplazy model, or sometimes called the 2-state perfect phylogeny model. And this is, if you think about it, essentially the most constrained evolutionary model you could have, right? And this red mutation right here on this particular tree actually follows this model, right? You can see it changed, this first character changed from 0 to 1 only once on the whole tree. That's all that happened with that character. It's a very, very simple model. All right, and these are essentially the 2 extremes. No constraints, any transitions are allowed with certain score or probability, or very constrained. The character has only one transition, only one change in state over the whole tree. Okay, yeah, question.
Once you have this set of characters 0 and 1, and here it's A, T, C, G.
Yeah.
What does 0 and 1 stand for?
Right. So this again would be So how do you model the specific character? So if you're thinking about a DNA sequence, right? There are a priori 4 possible letters we could have, nucleotides. So it seems natural to have a 4-state character. However, most of the time in many somatic evolutionary processes and even in population genetics, many loci that we see never exhibit all 4 of those states. So they might only exhibit 2 or 3. Right? If they exhibit one state, they're uninteresting for any sort of inference. So population genetics, sometimes biallelic SNPs, you might have heard that term, where there's only 2 of the 4 possible states. Okay, so why might we want to use a more constrained evolutionary model? Why do people actually do it? So this model I actually mentioned in population genetics is used. In cancer evolution it's used. And one of the reasons it's used a lot in cancer genomics and in single cell sequencing in particular is that by using a constrained model, we can avoid sort of overfitting the data when there's high rates of missing data and errors. Okay? So if we allow any transitions, then we sort of start conflating the error process and the mutational process. And if we think that, we have good reason to think that perhaps, that it's rare that a mutation would change back from 0 to 1. We make this infinite sites assumption. It becomes valuable to use this in our phylogenetic approach. Okay, so 2 states per phylogeny, widely used in cancer genomics. Finite states, widely used in species evolution. But it turns out there's a bunch of other, perhaps less well-known models that are kind of in between these two. There's a model called the Dolo model, something called the Kamen-Sokal model, and there's actually a few others. All right, and what I want to show you in the next several minutes is actually 2 other new models that we recently described for dealing with 2 different applications. One is tumor phylogeny, and this is going to be a model called the constrained K-Dolo model. I've already shown you here the state transition graph, so if that all makes sense to you, you don't have to listen to me describe it. And another is for normal development for experiments that induce mutations to track cell lineage. And because of the way these experiments work, this leads to a model that we dub the star homoplasia model with this state transition graph. All right, so let's sort of look first at the tumor phylogeny application. And this work was motivated actually by a very specific data set. So we're working with, and still are working with Chris Abuzio-Donohue's group at MSK, she's a pancreatic cancer expert, and she's done these amazing sequencing of all sorts of pancreatic cancer samples. And in particular, they've been using now some single cell sequencing technologies, and the technology they were using here is one from a company called Mission Bio. It's called the Tapestry platform. And what it does is it does what's called targeted single cell sequencing. So it doesn't sequence the whole genome, it sequences targeted regions of the genome, about 500 targeted regions, each about 200 base pairs. This is a PCR-based technique. But what you can get is really high coverage, read coverage, of those targeted amplicons in thousands of cells, all from the same experiment. Okay? So if you think about how we identify mutations in the genome, if I've got 50 reads covering an amplicon, I can very reliably call single nucleotide mutations, maybe sort of small indels. But because I've sequenced so little of the genome, I have less, I almost want to say power, although I don't have a statistical argument, but less power to identify these larger copy number aberrations. And actually, it's even trickier to do this because they're in the experiment, because it's PCR-based, you sometimes have whole amplicon dropout, things that actually look like copy number aberrations. So if one wants to build a phylogeny from this data, it seems to make sense to use a model that uses single nucleotide mutations as the phylogenetic characters. And since there's high rates of missing data and errors in these measurements, and since this is a tumor, and we think that maybe it's pretty unlikely that the exact same single nucleotide mutation will have occurred, same position, same mutation, during the evolution of this tumor. So if we make this infinite sites assumption, or this perfect phylogeny assumption, it's become a standard thing to do in cancer evolution, then we can start to try to reconstruct a phylogeny. So here's actually the data. This is like data seems so ridiculously silly because it's 7 mutations. Right? So when we build a phylogeny with these 7 mutations, and here they are. It's black here if it's present. So this is this mutation matrix. White, the mutation is not measured. Red here is missing, meaning like we didn't even have any reads covering that part of the genome, so we just don't know if the mutation was there or not in that cell. Okay? And so if we go run one of the state-of-the-art methods for doing this perfect phylogeny model, what we get is a tree that looks like this on these 7 mutations. All right, there it is. But what it actually starts to infer, because it's forced to make this assumption that mutations are gained only once, to fit things into a tree, it starts assuming that there's really high rates of false negatives in the data. Okay, so this like constrained model has forced these really high rates of missing data. So this is this mutation BRCA2. And what you can see is that this mutation, the way the tree is fit, is that it should be like in all cells. So what it's really doing is it's imputing this mutation in these cells for which it was not actually measured. All right, so one could ask, well, why maybe we should just go to the fully unrestricted model, and people have done that, and that solves some of the problems, but can introduce other problems. So what if we take a sort of step away from perfect phylogeny and sort of think about the process itself, and think about these single nucleotide mutations and the fact that we've allowed them to be gained only once. And it turns out that actually these mutations are lost frequently. They go back to the 1 state, but they don't go, from the 1 state, they go back to the 0 state. So 1 here is the mutation present, 0 is the mutation absent. But the reason they go from mutation present to mutation absent is not because the nucleotide reverts back. There isn't a mutation of that same nucleotide back. Instead, the whole locus, the whole region gets deleted, because deletions are very common in cancer. So if you think of this particular now diagram of here's now sort of a prototypical cell. We've got 2 copies of this chromosome. This is the 0 state, no mutation. Mutations introduced on one of the copies here. So now we're in the 1 state. And now if we remove this chromosome, it's lost, the one that has the mutation, then we go back to this other state, which looks to us, if we're just measuring presence or absence of this mutation, like the 0 state. Of course, it's not the 0 state. Something different has happened, right? So a mutation, single nucleotide mutations, it's a 0 state. If you think about these copy number aberrations, it's more complicated. So could we relax just a little bit this model of perfect phylogeny? And this is a model that's called the K-Dolo model. So what's happened here? We still assume that a mutation happens only once. It's gained only once in the evolution of the tree. But then it can revert back any number of times, and we allow some maximum here, k. So this is sort of modeling that there's going to be losses, gains only once. That's a mutation happening, but then because of these deletions that occur, can be lost many times. And so this K-Dolo model was introduced and used for single-cell sequencing by Mohammed El-Kabir, who's in the audience, and he had a method which I never know if I'm supposed to, is it sapphire or sphere, or how are you supposed to say that? Sapphire, all right, it's pronounced sapphire. And so we run sapphire, and it does something actually quite interesting. It says that all mutations are lost on the tree. So this is kind of interesting. There's only 7 mutations. They're all gained, and then they all just get lost in different cells, which seems strange and perhaps unlikely. And part of what's happening here is that the model, being more flexible, starts just allowing these losses to happen anywhere. This is what would happen also if you go to this finite states model. You just start getting a process that, because of the high rates of missing data and errors, start making those into transitions, mutations. Okay, hard to distinguish between the two. So when my postdoc, Palash, whose name and picture I'll show in a second, sort of showed this to me, and he said, hey, I want to work on a new model. And I said, wait, we've already actually solved this problem, because we published a paper a few years ago with a method that looks a little bit at where these copy number changes happen, these deletions and duplications, and allows these losses of the mutation only when there's data in the reads to say that there's been a deletion. So it sort of constrains where these losses can happen. We call this supported loss model. And so we did this, and the only problem was that we couldn't actually, with this targeted data, identify where the copy number aberrations were. So what Palash proposed was that we could do something sort of interesting and in between, which is we could allow these losses, but allow them only between certain groups of cells. Because what he noticed was that he could take the whole profile, all the reads across all the cells, and he could group the cells by their read counts into these coherent groups of cells or clusters, which are driven by these changes in copy number at different loci. This is a Tisney plot, sorry. It should probably be UMAP or whatever the current state of the art is for making these things. Fate map, who knows? But anyway, there's these three clusters. And so could we use actually this clustering to constrain where the losses would be? So here, if we have these three clusters, we would allow the losses to occur only between the clusters. And I think, in the interest of time, I'm just going to skip through these slides quickly to sort of say you can write all this down. It leads to this sort of interesting model where you've constrained these losses in a particular way. You've constrained them based on how you've clustered the rows of the matrix. Okay? And it sort of interpolates between the perfect phylogeny model where there's no losses, so you don't get very good phylogeny, it's sort of too cold, the soup is too cold. This K-Dolo model where losses can happen anywhere, and so they start to happen anywhere, and then you get losses that are too many, so it's right in between, sort of just right, and it seems to work pretty well. So there's a nice combinatorial formulation of this. You can write all this down, you can prove things about this model, you can show what the constraints are that you need to meet, and then you can wrap all these constraints into a integer linear programming. And this is what Palash Sashital, as a postdoc in my group, had done. He named this method Condor for the constrained Dolo reconstruction. Okay, so again, what it does is it takes the mutations here measured via the read counts, as well as a clustering of the cells, and then it builds a phylogeny based on the read counts of the mutations, as well as constrained by the groupings of the cells. All right. Works well. I'm not going to show you the simulations. And then in practice on this data, something quite interesting and we think sort of useful happens, because we can see that actually when we run on the 2,000 cells from these two different regions of the pancreatic tumor, we see a nice tree where it's well split between the two regions. This was not imposed. This is what happens. We throw all the cells together. But most of the cells from one region end up on one branch of the tree. Most of the cells from the other region end up on the other branch of the tree. And there's only 2 mutations that are lost. And in fact, these 2 mutations are in the same gene. So it's really just a single gene loss. So many fewer losses than what was obtained with this K-Dolo model, and much more reasonable than what's obtained with a fully constrained 2-states model. Now, we can also sort of run this on other data sets. You can actually sort of look at single-cell sequencing data sets of metastatic cancers. And when you have different trees for tumors where there's measurements of both primary tumors and metastasis, and when you build the tree, and then you look at where those cells were located, it tells you something about the migration process of cells between these anatomical sites. And so the original publication that had analyzed this metastatic colorectal cancer data set had found this polyclonal seeding, where cells multiple times left the primary tumor and went to the metastasis. And what we can do with actually our older method, but now recapitulate with Condor and a little bit better, see that actually there was really only a single event where cells left the primary tumor and seeded the metastasis in the liver. All right, now we're at 3, but actually we started a few minutes late, and I want to, if I can, take 5 minutes to show you lineage tracing. So you can see all this, a preprint. It's coming out soon in review. But let's look very quickly in 5 minutes at the second model. So here we're looking at developmental processes, right? So we'd like to just build the tree from cells at the present time that reconstruct. Wouldn't it be amazing if you could, reconstruct all cellular divisions from human, back to the original fertilized egg? That seems actually nearly impossible to do. However, it has been done for C. elegans, which has just 959 cells. A very laborious process that in the end resulted in a Nobel Prize to Sidney Brenner and others, where they constructed the whole lineage tree of C. elegans. And once you have this tree, then you can start looking at what the ancestral cell states are. You can start learning a lot about development based on being able to couple what you can measure about cell state along with lineage. Now, Doing this for human in the way they did it is just completely impossible, right? You can't directly measure the process as it goes. We can't sequence enough single cells with high resolution to do this retrospectively because mutations occur too infrequently naturally. So we'd have to sequence the whole genome of many cells in order to do this. So the approach that's been taken recently is to use genome editing technologies to induce somatic mutations at particular locations in the genome that then we can read out via single cell sequencing. And there's a number of different ways to do this. And our collaborator introduced one of these approaches based on the CRISPR-Cas9 system. And so what's done here is a lot of sort of details, but it's really just induced genome edits at particular locations in the genome. And because of the way CRISPR works, you build these guide RNAs at particular locations. Here are these cassettes. You introduce multiple cassettes in different parts of the genome. The guide RNA guides CRISPR to edit this part of the genome as the organism is developing, and that edit will result in an indel. And you can get many, many different indels that occur at these particular locations. But once the indel, insertion or deletion, has happened, the guide RNA is no longer present, and so the mutations are irreversible. You never get a mutation at an edit at the same location again. And moreover, there's actually another process. Not only does the mutation not revert to the ancestral state, but you can't introduce a new mutation at the same location either. So this is what's called non-modifiable. So now our evolutionary process has many states because you have many different edits at a particular location. It can't go back to the ancestral state, but it also can't change once it's reached a particular state. So now we can think about these state transition graphs and what this should look like. And I should say that the other cool thing about this is that the readout is single-cell RNA-seq, which is cheap, so you can get this from many cells. So you get this readout, and you try to revert back. And the data is really messy. Okay? There's large numbers of possible states because you can get many, many different indels. So if we think of a phylogenetic character, instead of having 2 states, 0, 1, or 4 states like ACTG, you could have 50 to 100 different states at a particular location. There's many missing entries, and they have this special property. So what's the evolutionary model that could capture these types of characteristic features? And if you think about the usual finite states model where nothing is constrained, certainly you can do this model with as many states as you want, but it is not irreversible, right? Every transition could happen, and it's not non-modifiable. It's inherently modifiable, right? Any transition can happen at any time. So this is not the state transition graph that we need. We need something more constrained. And if you think for a minute about what these properties should be, what you end up getting is a graph that looks like this, where this is the ancestral state. Before CRISPR edits this site, CRISPR can edit it into, this is the one. CRISPR can edit it into any one of these possible derived states. But once it's done that, that's it. There's no more edits at that location. So this is what's called a star graph. And so this leads to what's called the star homoplasy model. Homoplasy because at different locations in the tree, the same location could be edited. Just once it's edited in a cell, it's done. So this is actually quite fun and interesting because how do you compute this model? And can you compute this model? Could you actually look at one of these matrices, which now has 0, 1s, 2s, 3s, 4s, up to the number of states? Could you look at one of those matrices and tell me whether it was a star homoplasy matrix? Does it meet this model? It actually turns out to be a hard problem. And then, given that, could you find the tree? So it's a hard problem, but again, it's got this nice combinatorial structure. And now I'm almost at 30 minutes, so I'm out of time. So I can't tell you the model, but you can write it down. You can relate it to other models. In particular, you can relate it to this perfect phylogeny model in a very interesting way. And then you can, again, derive a combinatorial algorithm to solve this model in the maximum parsimony setting. And then if you do that, you can apply it, and you can find actually interesting things in data. And I'm just going to show you one quickly. This is probably one of the largest lineage tracing studies where this was CRISPR editing in a mouse model of cancer. So they had induced cancer in these mice. They then not only were sort of measuring mutations in these, which is interesting, but they were also measuring lineage. So then you could, from these different sites where there was cancer and the cancer spread to different metastatic sites, you could measure the lineage. You could build a tree on all 20,000 cells. So here's the published phylogeny, and here's our phylogeny. You can't really see much from this because it's at least 20,000 cells. But the interesting thing about the phylogenies, one, ours is more parsimonious because we used a model that actually models the CRISPR editing process. So it's a more parsimonious model. This was sort of, that's what the objective should have been optimizing. So that's sort of a good thing. But it actually says something different about the metastatic process, because when you use the different tree and then you look at how many times did cells migrate between these metastatic sites, you get a different result when you have a different tree. So you actually get a more parsimonious explanation of the metastasis. All right, so that's sort of the summary of this one. Also a preprint, and this was at Recom earlier this year, and it should come out soon in Cell Systems, we believe. So that's the sort of summary. Very different, no statistics, a lot of combinatorics that I didn't show you, but sort of playing this game of tailoring your evolutionary model to the particular features of your data. And rather than use sort of the generic model, if you use a model that's more specific to the features of your data, you can actually get, in some cases, we think, better results. So I'll end there. There's the folks that have worked on some of this and our collaborators for the cancer part, and Michelle Chan for the lineage tracing part. Thank you.